So we're going to end this chapter with this one last section on phenotypic plasticity. The previous sections have focused on the genetic components to the phenotype and the variants in the phenotype. This last section is focused on the environmental component that influences the phenotype. And so that environmental component, you might remember from earlier sections in this chapter, is designated as V sub E, the variance that's attributable to the environment. And so now the environment includes all external factors that influence development. So anything having to do with light, water, nutrients, all kinds of stuff. So as an example, the figure on the right-hand side of the slide here is, showing you how the environment can affect the phenotype in a plant. Now, plants that are genetically identical, plants that are basically the clones of one another. So by using organisms that are bred to be essentially clones, we can expose those organisms to different environmental conditions and see what kinds of variation there might be in the phenotype given environmental variation. And what that'll do is it'll show us the degree to which the genetics of a particular genotype is malleable and can express different phenotypes from that same exact genetic makeup. So my recording image on the top right of the slide is over two different plants. The one on the left is a small plant and the one on the right is a large plant. And those plants resulted from being grown in different amounts of light. So low light conditions gave the small plant, the high light conditions gave the large plant. And specifically for the graph that's shown right in the middle of that figure, total plant biomass is what's shown on the y-axis. So under low light conditions, plants with this particular genotype have relatively little biomass. Plants that are grown the exact same genotype, keep in mind, under high light conditions, more biomass. Okay, straightforward enough. Now, what happens in terms of leaf size if these plants are grown under different conditions? So leaves, of course, are the photosynthetic organs of plants. So if we take these genetic clones and we grow them under low light conditions, then leaf size tends to be relatively big. Plants are investing energy into having bigger leaves as their solar panels to capture that light. The exact same genetic blueprint of a plant grown in high light conditions grows small leaves. So this is showing phenotypic plasticity. This is showing the capacity for one individual genotype to express more than one phenotype, depending on the environment. That is the definition of phenotypic plasticity. Now, there's a range of responses that might occur to the environment, and those range of phenotypic responses are referred to as the reaction norm. So we can figure out the reaction norm by plotting, such as on the figure on the lower left, a range of environmental conditions on the x-axis. So in this particular case, low light to high light. And then the phenotype that represents a response to those different light conditions on the y-axis. In this case, the phenotype that's being observed and documented is the leaf size. So you can see there's sort of a line there, a purplish line, low light, big leaves, high light, small leaves. And so that shows the reaction norm. Actually, the reaction norm can be seen with the blue lines going across horizontally, you can basically predict what the leaf sizes are going to be based on that purple line reaction norm. So there's predictability to this once that you conduct an experiment and figure out the reaction norm. So as I've already mentioned to you on the slide, the way that you study phenotypic plasticity is growing genetically similar organisms under different conditions, like the plant example on the right is showing you, and the graph on the bottom corner. So by doing that, the variance in genetics is 0 if every plant is genetically identical. And that means that the variance in the phenotype, V sub P, will be entirely equal to the variance due to the environment. So this is how we can isolate the effects of the environment on phenotypic variants, is using genetically identical organisms and exposing them to a range of environments and then figuring out the reaction norm. Now, sometimes different genotypes respond differently to the same environment. And in this case, reaction norms can vary from one genotype to another. The interaction of genes and the environment is a whole nother component of phenotypic variants that you haven't been exposed to before, and you're being exposed to it on this slide. So far in this chapter, we've dissected down phenotypic variants as including those components that are additive, genetically additive, those components that are due to dominant recessive interactions, those components to the phenotypic variants due to epistasis, and on the last slide, introduced how we can determine the component of the variance that's due to the environment. But now we have this last expression, this fifth expression of what can contribute to the overall phenotypic variance, which is the interactions between genes and the environment. And so that's V sub G by E, which you can see there on the slide. So as an example, scientists did some studies on nematode worms, and they exposed worms to different temperatures. You can see in the upper figure A, temperatures ranging from 12 to 24 degrees Celsius, and then documenting the age maturity for worms under those different temperatures. And as you can see in figure A, at low temperatures, all the worms tended to have a older, longer time till age maturity, whereas the higher temperature experiments had a younger age till maturity. So this isn't showing really any genetics by environment interaction. However, if you look at figure B, which is fertility, okay, number of offspring, number of eggs specifically, then when worms were exposed to the same range of temperatures, but then their fertility was examined, you can see that there's lines crisscrossing all over the place. So what that's telling us is that genetically different worms being exposed to the same environmental conditions are showing different phenotypic expressions. So this is telling us that there is a genetics by environment interaction when it comes to fertility, but there isn't a genetics by environment interaction when it comes to age at maturity. So on the very bottom left of the figure in part C, it's showing, okay, once this interaction was found between fertility and temperature, then they were able to do a Qtl analysis, again, from a couple sections back here, doing inbred lines of worms, for different phenotypes, then crossing them 2 times to get F2 hybrids, and then from those F2 hybrids, the steps of a Qtl analysis from those F2 hybrids, figuring out which genetic markers in the chromosomes are related to which phenotypes. And so you can see that scientists were able to do a Qtl analysis and identify a region on chromosome 4 that can help explain this interaction. So whereas the last slide was specifically telling you how we can determine just the environmental component on phenotypic variants, what this is showing you is that it's also possible to have a genetics by environment interaction that contributes to variance, and we can pinpoint that as well by doing these sorts of breeding experiments and plotting the results. So now you know what phenotypic plasticity is, the range of phenotypes that are possible with a particular genetic makeup when that genetic makeup is exposed to different environments. But can phenotypic plasticity evolve? Well, the answer is yes. Phenotypic plasticity can evolve when the variance due to the genetics by environment interaction is greater than 0. If that component is greater than 0, then plastic responses can evolve. One key experiment that demonstrates this is shown here, this big figure graph of fruit fly thorax length over many generations. So body size in fruit flies, Drosophila, is influenced by temperature. And so siblings, of course, that are genetically similar, were reared at temperatures of either 19 degrees or 25 degrees. And over 22 generations, phenotypic plasticity increased in flies, in the line of flies that were bred with the biggest differences in body size. And so that's shown by the green line. The green line represents those flies in a particular genetic line that were bred, the biggest differences in body size over the course of 20 generations. And the lines that are shown in blue shows the results of breeding flies in a separate line that had the smallest differences in body size. And what we can see is that the flies, the populations did evolve. Plasticity increased in one of the lines, in the green line, and it decreased in the other line. So yes, indeed, phenotypic plasticity can evolve. And again, it can evolve as long as there's a genetics by environment component of the variance that is greater than 0. So here the key question is, can phenotypic plasticity evolve? And the answer is yes, as long as the genetic by environment component of the variance is greater than 0. That's the answer. And if that component is greater than 0, then yes, plastic responses can evolve. So here is demonstrated, being shown to you, the results of an experiment where fruit flies were exposed to different temperatures to see, to investigate the evolution of phenotypic plasticity. So we already know body size in Drosophila and fruit flies is influenced by temperature, so that is known. So what was done here is siblings were raised at either 19 degrees Celsius or 25 degrees Celsius. So, of course, the siblings are genetically similar. So siblings from different families were raised at either 19 degrees or 25 degrees. Then what the biologists did is they took those flies from each of the families that showed either the greatest difference in body size when exposed to these temperatures, and so that's represented in green. So those flies that showed the greatest differences range in body size at these temperatures were the most phenotypically plastic. So they wanted to know, can we increase the phenotypic plasticity? Can we cause the phenotypic plasticity to evolve? So they took those flies and bred them, the ones that had the biggest differences in body size. And lo and behold, over 20 generations, the plasticity of the body size, as represented by the thorax size, did increase. The green line was going up. They also took flies that had the smallest differences in body size, the least phenotypic plasticity when raised at these different temperatures, and they bred those over 20 generations, and that's represented by the blue lines. And in those groups, the amount of phenotypic plasticity indeed decreased. The gray lines are controls, which are just random flies that were grown under the same conditions. And you can see those gray lines are more or less in between the blue and the green ones. So the outcome of this experiment shows that phenotypic plasticity did increase in one line, the green one, decreased in the other line. So we have empirical evidence based on these fruit flies that indeed phenotypic plasticity can evolve. So here's a quick final note about phenotypic plasticity and bringing it into the context of climate change and how species might respond or not to climate change based on their phenotypic plasticity. So here we're looking at snowshoe hares. In the summertime, they grow brown hair, blends them into the environment. It's adaptive. In the wintertime, they change their hair out. It's mostly white, blends them in with the snow, adaptive, difficult for predators to see them. So, but with climate change happening, winters are coming later. Spring is coming earlier, so there's less snowfall, there's less snow around. And the snowshoe hares are now becoming mismatched to their environment because they're changing their color patterns and their hairs in a typical cyclical way, as they're genetically programmed to do with this phenotypic plasticity. But with climate change happening, now they're out of sync with the snowfall and the snow covering the ground, so it's leaving them more vulnerable to predators. So what's going on here is how rapidly populations can respond to climate change depends on factors such as heritability of coat color change, so the heritability of phenotypic plasticity. So in the last slide it was shown fruit flies, absolutely, their phenotypic plasticity can evolve. So depending on the component of the phenotypic variants in these snowshoe hares, that is determined by that genetics by environment interaction. If there is a significant component to that, remember, as long as it's greater than 0 from the last slide, phenotypic plasticity can continue to evolve. And so it remains to be seen for any species out there that respond to climate change. How they're able to do that might depend on their ability to evolve their phenotypic plasticity.